Species concepts, part of the Systematics and Biodiversity course offered by Leiden University in 2020. I'm Jeremy Miller. In this lecture, we will explore some of the ideas about what species are and how scientists recognize them. We may say that taxonomy is the scaffolding that all of biology is built upon. We are self-evidently surrounded by an amazing diversity of life forms and processes. As we observe our world, we may recognize certain individuals that together share particular characteristics that allow us to recognize them as distinct from the various other kinds of things that we encounter. We call these different kinds of things that we encounter and observe in the real world species. And every year in the modern era, we publish descriptions of about 17,000 new species. But when we dig a little deeper, we find that coming up with a definition of species is not so straightforward. One recent review reckoned that more than 24 different species concepts could be found in the modern literature. It's probably fair to say that the most widely known and discussed of these is the biological species concept, proposed by Ernst Mayr in 1963, which is built around the concept of reproductive isolation. If members of two populations can produce viable offspring, they are the same species. Otherwise, they are different. Despite the popularity of the biological species concept, it is clearly not a universal one. For one thing, it ignores asexual reproduction. For another, its real-world application rarely has much to do with mating, but instead relies on other characteristics, typically clusters of morphological similarity, as a proxy for reproductively isolated units. We rarely perform breeding experiments in the pursuit of taxonomic questions, and of course, such a study would be completely impossible in the realm of paleontology. So maybe this is all a bunch of nonsense. We can't seem to agree on the fundamental question of what a species is. So if taxonomy is a science, then the things that taxonomy describes, species, must be independently testable. Without a clear definition of what a species is, how can we test our conclusions? If true, this has some pretty profound implications. For example, if there is no such thing as a species, how could there be such a thing as an endangered species? What does it mean to devote resources to protect endangered species if they are, in some sense, not real? In the world of philosophy, this topic is typically discussed in the context of natural kinds. There are many ways to sort objects into different categories using arbitrary or artificial criteria, but the concept of natural kinds means that there is only one natural way to group the entities being classified. The fact that species are mutable over time basically disqualifies them from being natural kinds because investigations conducted at different times may not be looking at the same reality. If species are not in some way natural kinds, then differences of opinion within taxonomy are simply that, disagreements. But if taxonomy is a process of discovering something real about nature, then disagreements should be possible to resolve decisively, given the right data. We have some examples involving things like medicinal plants and birds, where traditional folk taxonomies of non-Western cultures are identical to those produced by scientists in the Linnaean tradition. The existence of two very similar independent classification systems tends to support the idea that taxonomies are getting at something real in nature. Even before Darwin and Wallace proposed evolution by natural selection, it was understood that species could change over time. Jean-Baptiste Lamarck, in his 1809 Zoological Philosophy, described the transmutation of species, proposing that a species could change over time. This contrasted with classical ideas about nature, which supposed that species are distinct and unchanging. Darwin suggested not only that populations could gradually change, but that one species could split into two, that over time, sufficient changes could accumulate to the point that a new species has formed. But way back in the mid-1800s, Darwin was frustrated by the multiple and inconsistent ways to conceive of species. It did not seem that any one of these concepts was completely adequate in all cases, and the choice of criteria had implications for what was or was not considered a species. Furthermore, the boundaries used to define species across space and time were similarly tainted by arbitrariness. Sir Joseph Dalton Hooker was a British botanist and a friend of Charles Darwin's. In a letter from Darwin to Hooker in 1856, Darwin articulated some of his concerns about the species concept. He wrote, "I have just been comparing definitions of species and stating briefly how systemic naturalists work out their subject. It is really laughable to see what different ideas are prominent in various naturalists' minds when they speak of species. In some, resemblance is everything, and dissent of little weight. In some, resemblance seems to go for nothing, and creation the reigning idea. In some, dissent the key. In some, sterility an unfailing test, with others not worth a farthing. It all comes, I believe, from trying to define the undefinable." A major turning point for evolutionary research occurred in the 1930s, when Fisher, Haldane, Wright, Dobzhansky, and others developed mathematical population genetic models to illuminate the genetic mechanisms of evolutionary change. This school came to be known as the modern synthesis. Julian Huxley coined the term in his 1942 book, Evolution, the Modern Synthesis. The integration of genetics with models of natural selection shed tremendous light on and strengthened Darwin's views on evolution. Here was the missing mechanism that introduced new variation into populations via mutation and recombination. Indeed, thanks to the modern synthesis, much of current research in evolutionary biology is strongly tied to genetics, and current methods for studying speciation are no exception. The modern synthesis led to advances not only in the study of evolution within populations, but also changes in the way species were defined and in how new species were considered to form. Under the commonly used biological species concept of Meyer, the formation of new species involves the evolution of reproductive barriers to the production of viable offspring, either before or soon after mating. Barriers that prevent mating are referred to as prezygotic barriers, while those that operate after mating are called postzygotic barriers. Thus, new species form when individuals from diverging populations no longer recognize one another as potential mates, or opportunities for mating become limited by differences in habitat use or reproductive schedules. In some cases, these prezygotic isolating mechanisms fail to prevent interbreeding among individuals from separate populations. In such cases, viable hybrids may form, or mating may produce inviable zygotes or sterile non-reproductive hybrids. These diverse pre- and post-zygotic barriers are of great importance for understanding speciation because they determine how reproductively isolated populations are from one another, which indicates how far along the often continuous process of speciation a population is. For example, reproductive isolation is weak in the early stages of speciation, but changes to strong or complete in later stages of speciation. One or more of the many types of isolating mechanisms may come to play a role in the evolution of species along a continuum. Because the concept of isolation is central to the biological species concept, theoretical work concentrated on mechanisms for isolation and the consequences for species formation. Geographic isolation, such as the formation of a new barrier like a mountain range, or dispersal of propagules to a distant island, is perhaps the most intuitive, but proponents also conceived of isolation in the wake of ecological, behavioral, and genetic shifts. But the biological species concept did not fully address Darwin's worries. Reproductive isolation was recognized as not simply present or absent, but as a continuum where the degree of reproductive isolation would be reflected in the degree of genetic or phenotypic distinctiveness. At either end of the continuum, species boundaries are clear, and different taxonomic experts are likely to recognize the same species boundaries. But within the intermediate zone, species boundaries are not well defined, and experts may disagree in ways that cannot be resolved using unambiguous criteria. Within this intermediate zone, daughter lineages may accumulate evidence of their distinctiveness. For example, they may become morphologically distinguishable, reproductively incompatible, or ecologically or behaviorally distinct. Ring species exhibit a form of incomplete reproductive isolation that presents challenges for the biological species concept. Ring species consist of a number of populations geographically situated in such a way that adjacent populations can interbreed, but some more distant populations cannot. Reproductive isolation suggests that more than one species is present, although gene flow, mitigated through intermediate populations, is possible throughout the complex. There are several real-world examples, including Ensatina salamanders in California. Another idea was articulated by George Gaylord Simpson. He said that something even more fundamental about species than Meyer's idea of shared reproduction is at work, that the members of a species have shared an evolutionary process and an evolutionary history. Simpson's key idea is that a species is an evolutionary lineage that has evolved separately from other species. In other words, the organisms within a species all share in the process of evolution. The process of evolution, including genetic drift, migration, and adaptation, caused there to be a thing, an entity, made up of organisms evolving in concert, and that collectively form a species. Importantly, this property of evolving together does not apply to kinds of organisms above the species level. For example, birds are not all evolving together. There are many separate species of birds, each of which is on its own evolutionary path. The same goes for mammals and plants, and indeed any kind of inclusive taxon that includes multiple species. The nice point about the general idea of a species as an evolving unit is that it fits those organisms, such as many bacteria and some eukaryotes, that do not engage in sexual reproduction. The related phylogenetic species concept of Joel Craycraft builds on the foundation of Simpson, integrating advances in quantitative phylogenetic analysis. Issues such as asexual reproduction and horizontal gene transfer present challenges to the species concept debate, particularly the biological species concept with its focus on interbreeding. But it is worth pointing out that many organisms that reproduce asexually do not do so exclusively, meaning that normal genetic exchange between conspecifics still occurs, even if not with the regularity of species that exclusively reproduce sexually. Prokaryotes, archaea and bacteria, reproduce asexually through binary fission, in which the parent organism divides in two to produce two genetically identical daughter organisms. Eukaryotes, such as protists and unicellular fungi, may reproduce in a functionally similar manner by mitosis. Most of these are also capable of sexual reproduction. Prokaryotes are also groups with limited morphological characteristics. Horizontal gene transfer means that a single organism may have components with independent evolutionary histories. In practice, prokaryote species are typically defined by their molecular distinctiveness from similar species based on either DNA hybridization or sequence similarity of a standard marker. Remember that methods of phylogenetic reconstruction allow us to plot the history of characteristics on a tree. Distance-based methods do not allow this, and while they are computationally very fast and may approximate evolutionary relationships, the most closely related species are not necessarily the most similar. We often use a prescribed amount of sequence difference, like 3% sequence dissimilarity, as a cutoff for what is considered within species variation as opposed to species-level divergence. But while we may be able to use knowledge of the evolution of a taxon to come up with a distance cutoff as a heuristic for finding species limits, this approach does not escape arbitrariness or resolve ambiguity across the speciation continuum. Hybridization presents another challenge for species concepts. Under artificial conditions, we can cross animals that would not reproduce under natural conditions. Such novelties present a challenge to the biological species concept, but we typically associate hybridization with poor species health, particularly in the context of conservation efforts. But hybridization may sometimes play an important role in diversification. For example, a recent study of cichlid fish in some African lakes found that fish from Lake Bangwele colonized Lake Meru, where they hybridized with Congolese lineages and then underwent multiple adaptive radiations with striking ecological and morphological consequences. This is very different from the classical model of adaptive radiation, which was thought to require genetic isolation and lack of competition from related species. As with lateral gene transfer in prokaryotes, the evolutionary history of these fish will not be tree-like. Polyploidy is a condition in which the cells of an organism have more than two paired sets of chromosomes. Most eukaryotes are diploid, meaning they have two sets of chromosomes, one set inherited from each parent. Polyploidy may occur due to abnormal cell division and is particularly common among some kinds of plants. Polyploidization can be a mechanism of sympatric speciation because polyploids are usually unable to interbreed with their diploid ancestors. Some estimates suggest that 30 to 80% of living plant species have polyploidy in their evolutionary past. In practice, the gold standard for progress in taxonomy is the taxonomic revision. This typically involves a study of all species in a particular taxonomic group, say a genus or small family, from a major geographic area or the entire world. This entails reviewing all specimens from the world's major museums, as well as all the previously published taxonomic literature. Each specimen is associated with data about where and when it was collected. By studying the characteristics of these specimens, all these specimens are sorted into a number of species that can be distinguished from each other. The characteristics involved typically include morphological features, but may also include behavior, DNA sequences, and other kinds of characters. Each of these species has either been described previously or not, meaning it is new to science. Some of the specimens in all these museum collections were used by earlier authors to describe species. In most cases, particular specimens have been designated by the original authors as the type specimen, that is, the voucher specimen tied to a particular species name published at some time in the past. Some of the species in your global taxonomic revision will have been described previously, possibly several times. The rules of nomenclature help to determine which names should be applied to such species. The rest receive new names. Because these specimens were studied in a comparative context, the species identification of each specimen studied is often the most accurate available. It's easier to get it right when you have specimens representing all the species in the world of a particular taxon available for direct comparison than it is for a taxonomist using a publication to identify a particular specimen. The mechanisms of scholarship, particularly the citation of specimens used in the study, are key because this is the mechanism by which other workers can subsequently revisit and reevaluate the particulars of the species delimitation presented. Taxonomic treatments typically include a diagnosis section, which describes how to distinguish one focal species from all others. This is key in the sense of taxonomy as a scientific endeavor, because this is where we will find errors or omissions if there are problems in species circumscription. Remember that in science, we want to make bold and testable statements based on evidence. So, for example, if I establish a new species, either I'm right that this species has not been described previously, or I'm wrong and it has been. If I write in my diagnosis what sets my new species apart from all others, and yet another description exists for the same species, then there is something wrong with my diagnosis. Scientific progress depends on someone discovering and correcting this error. Even skilled and careful workers can make mistakes at this stage for several reasons. For example, many species come in multiple forms that may look different from each other, such as occurs with sexual dimorphism, in which case it may not be clear which males are associated with which females. But additional collections or molecular data might eventually lead to refinements and improvements in the circumscription of our species. The problem with species concepts may be related to the prospect that we have confused how we conceptualize species from the way we recognize them. Some species concepts, like the biological species concepts, are rooted in process, in this case, sexual reproduction. Others, such as the evolutionary species concepts of Simpson and related concepts that followed, conceive of species within the context of evolutionary history. But this is distinct from whether or not someone might be able to recognize species or species boundaries in a given situation. By contrast, concepts based on similarity, including morphology or genetic distance, can be thought of as more or less practical heuristics. When we observe nature, we find clusters of similarity that are the products of evolution. We call these things species. But imagine what would happen if we had perfect knowledge of the morphology, behavior, and genetic content of every individual organism that has ever lived. Wouldn't the gaps between these clusters be obscured? Species are the things that are mostly between the gaps, and because species are mutable, the clusters of similarity that we observe reflect a particular moment in time. Evolution has rules. Probabilities of gene flow are regulated by phenomena such as natural selection, sexual selection, and interactions with other species in the environment. Species don't have rules. They are emergent properties of the process of evolution.